bond is a I share electrons bond. It's a covalent bond. What's the other kind of bond? Ionic. What results in an ionic bond? What are positive and negatively charged ions? Yes? And what determines the reactivity of an atom or element? Valence electrons. Everybody knows what a valence electron is, yes? How many valence electrons does hydrogen have? 1. How many valence electrons does helium have? 2. Yes? Good. I know. Space shot, I told you. Everybody understands patterns of chemical reactions, synthesis reactions, decomposition reactions, replacement reactions, yes? How about an exchange reaction? Well, that's yeah. So what do I do here? I take a phosphate from this guy and I attach it to the glucose. I exchange something within the reaction. Remember when I wrote up on the board AB plus CD equals C plus BD? Did you have a question? Good. Everybody else? Everybody good? So what influences chemical reactions? So I increase the temperature, it'll go faster. Why? The molecules are going to move faster. If they move faster, the chances are they're going to what? Bump into each other faster at a faster rate. So what happens when I decrease temperature?
They slow down.
Slow down reaction rate. How about concentration? So I increase concentration, I speak chemistry. Yeah, increase reaction rate. Yes? I decrease concentration, I decrease reaction rate. Good. Good. So we talked a little bit about water and dissociation of things in water. Water, because of its polar qualities, is going to help carry things around in solutions. Now here's the fun part. Acids and bases. Definition of an acid is the concentration of hydrogen ions in solution. Yes? Hydrogen ion, what's that? Yep. Yes? H plus and OH minus. So when water comes apart, that's what it comes apart into. Now when I add things to water that cause it to come apart and maybe have a higher concentration of these guys than these guys, hydrogen ions in solution than hydroxide ions in solution, I now have a solution that we call acidic. And that has a certain concentration of hydrogen ions in the solution, including the water that might come apart as well. You following me? Okay. So if I look at a solution that has an equal concentration of both of those guys, hydrogen ions and hydroxide ions in solution, I'm looking at this guy here. A pH of 7. That number represents the concentration of these guys in solution. Now if you look at this chart, OH on this side, hydrogen on this side. This is a minus exponent. So a -2 is actually smaller than a -1. Yes? How come? Which one's bigger, -2, -1? -1 is bigger. Why? It's closer to 0. Yes? Okay. So on this chart, and that's why I point this out, the number that represents pH defined as concentration of what? Hydrogen ions in solution. That number, pH, the number, is the concentration of hydrogen ions in solution. So when we look at a pH 7, yes, the concentration of hydrogen ions in solution is 10 to the -7 moles per liter. Now, in this case, it also happens to be the concentration of hydroxide ions in solution as well. What do we call that pH? It's neutral. Okay? Watch this. pH 9 is pH 9 because the concentration of hydrogen ions in solution is 10 to the -9 moles per liter. But what's the concentration of hydroxide ions in this solution? Which is what? It's more than the hydrogen. This is a basic solution. If I go to the bottom of the scale, when the hydrogen ion concentration is higher than the hydroxide ion concentration in solution, what kind of solution do I have? Acidic. Do you see what the pH means? It basically is following the strict definition. Concentration of hydrogen ions in solution. But when we look at a solution that is greater concentration of hydrogen ions in solution, the pH number goes what? Down. When we're equal, that's neutral, pH 7. And when we go higher, we're actually going lower in hydrogen ion concentration and higher in hydroxide ion concentration. That's basic. Now that can be a little confusing sometimes. So I look at the picture in your book. The picture is on, this picture is on, where is it? Page 40? Oh, good. So look at that for a while. Understand what pH is. One of the very important things that we're going to talk about when we talk about chemistry in the human body is maintaining a proper pH. So that concentration of hydrogen ions in solution is going to have an effect on chemistry on the whole. We have to maintain a very strict pH in our bodies. Does anybody know what normal body pH is? It's very, very tight. 7.35 to 7.45. So it's slightly what? Very slightly basic. That pH, that concentration of hydrogen ions in solution is going to affect a lot of our chemistry. So acid-base concentrations we are going to revisit many, many times in this class. We're going to talk about a disruption in pH and how it can affect different systems. So that's why we kind of have to understand what pH is. In order to maintain that fine balance with respect to hydrogen ions in solution, we have chemicals in our body that are going to take up excess hydrogen ions or give off hydrogen ions when they're needed. And these chemicals are called buffers. These buffers are described in our book on page 41. And we're going to talk about them when we talk about the respiratory system, and when we talk about the digestive system, when we talk about blood. We're going to talk about these molecules again. But these buffers, again, are very important chemicals that are either going to take up excess hydrogen ions or give off hydrogen ions if the pH starts to fluctuate. They're going to help neutralize different substances after chemistry has taken place. So buffers, buffer systems are described in your textbook, and we will talk about them again throughout this course. So acids have what? They have a low pH and they have a higher concentration of hydrogen ions than hydroxide ions in solution. Bases? Higher pH? Higher concentration of hydroxide ions than hydrogen ions. So if I take in this same beaker, dump hydrochloric acid into it, guess what's going to happen? Inside the beaker, I'm going to have H2Os. They're going to hang out. Get busted up into these, and that's definitely going to get up into this. Do you see what's happening in my beaker? I'm getting a higher concentration of who? Hydrogen ions in solution. So if there's more of these guys than there is these guys. Yes? So what kind of solution is that? Acidic. If I add this to the beaker, sodium hydroxide, what do you think is going to happen? Yeah, same thing, except I'm going to have more what? More of these OHs floating around, and that's going to give me a basic solution. Okay? Good on pH? Understand the difference between an acid and a base? Know what a buffer does for me for a living. It's going to help keep the balance. Because remember, I want to keep that concentration, 7.35 to 7.45 moles per liter of hydrogen ions. Otherwise, I start screwing up my chemistry. And that's the most basic I can tell you. Yes? Or acid, I can tell you. Questions? We good? All right, the next part of the book talks about organic compounds. Take the atoms and the elements and I stick them together, and they're going to react based on their electrons, and hydrogen ion and hydroxide ion concentrations are going to have an effect on that. My goal is molecules that I'm going to use in the body. Not only for building blocks, but molecules that I can use to break apart for different reactions of metabolism to make energy, for example. So organic chemistry is what the book talks about next. What do all organic molecules have in common? They have carbon in them. So what is an organic molecule? No. What is it? It's inorganic. Is sodium chloride an organic molecule? No. It's an inorganic molecule. Is carbon dioxide an organic molecule? Yes. It's got carbon in it. See? Carbon. What's it say in your notes? I'm not. They pile them in the organic, in the inorganic, but I put them in the organic category myself. So anything with carbon. Carbon is what? Organic. So inorganic chemistry larger molecules. That's probably why they stick them still in organic, because they're a little teeny. But methane is organic, CH4. Yes? There's groups that we have to remember in organic chemistry that are extremely important for us as humans and most living organisms that we know of. There are carbohydrates, there are lipids, there are proteins, or called nucleic acids. So what we have to understand in this chapter is basically what they're made of, and then what we're going to use them for in the human body. So the first group of organic molecules we're going to talk about are called what? Carbohydrates. Now the very smallest building blocks of carbohydrates fall into this category. C6H12O6 or C5. But you know who this is? Who's this? Is that small, by the way? That's glucose. C6, 6 carbons, 12 hydrogens, 6 oxygens. That's glucose. That is what we call a carbohydrate monomer. So when we think of these molecules, think of them as sort of like chains of things. Like, there's a whole bunch of pearls on a chain to create the necklace. When I look at carbohydrates, they can be simple or they can be complex. So I can have one, and what do we call one? A monomer or simple sugar. Or I can have a whole bunch that I hook together chemically. Now, how am I going to hook them together chemically? This is where our knowledge of chemistry comes in. If I want to bring two monomers, two simple sugars together, how do I put them together? No, I'm going to suck up the water. That reaction, linking two monomers, like two glucoses, for example, or a glucose and another simple sugar, like you probably have some in there. What's in there? Mocha Frappuccino. There's lots of them in there. Do you know what's in there? Chemistry? There's caffeine in there, but think of the carbohydrates. Sugar. That white stuff in the bowl. What is it? It's sugar, but you know what it is chemically? No, it's actually two simple sugars linked together by this reaction, dehydration synthesis. It's glucose and fructose. That in there is sucrose. So when I link two together through dehydration synthesis, I have what we call a disaccharide. Sucrose is a disaccharide. What if I link a whole bunch of these guys together? I make a polysaccharide. Poly means many. So in the carbohydrate family, I can have building blocks, and there's a whole bunch of different ones. There's glucose and fructose and galactose and ribose. Those are all simple sugars in the family. And linked them together in many different ways, I can create polysaccharides. Now, if I take my sucrose and I put it in my mocha frappuccino, and I stir it up, what happens to it? It dissolves, or use a chemistry word for dissolve, dissociates. If I add it to my frappuccino, which is mostly what? Well, yeah, in that case it's water. If I add water to a disaccharide, what do? I break it apart. Water, lysis. Hydrolysis reactions break apart what? Well, no, I add water to break apart, in this case, that disaccharide into monosaccharides. Okay? So my sucrose in there, floating around in the water, is actually glucose and fructose. Yes? If I want to make sugar again, what do? Suck out the water. Do you see the difference? Okay. So this is an example of a reaction, dehydration synthesis reaction of sucrose, and its breakdown by hydrolysis. So this is the fancy way of saying everything I just said. So I take glucose and fructose, and if I suck water out, I'm going to create who? Sucrose. That's the stuff I'm finding in the bowl. But if I take a scoop of this and I throw it into water and spin it around, add water to it, what am I going to get? Glucose and fructose, dehydration synthesis, hydrolysis. This is a what kind of reaction? It's a reversible reaction, depending on how much water I have, right? Everybody with me? All right, so everybody knows what carbohydrates are made of. Carbohydrates are made of sugar. Here are some examples of the simple sugars in the family, carbohydrate family. The difference between them is the number of carbons. Some of them are hexose, some of them are pentose. What the heck does that mean?
5.
Some are 5, some are 6. And where everything is on the molecule, spatially. These two sugars have the same chemical formula, C6H12O6. Are they the same? Why? They're not the same because of where all their other little functional groups, things hanging off the carbon chain, sit. That makes for a different sugar. Okay? So, this is dextrose is its other name, and this is from fruit, fructose. Both C6H12O6, but different because of where their functional groups are. If I hook them together, I can make different disaccharides. We already met sucrose. If I hook together a fructose and a glucose. Maltose. Anybody ever have malted milk balls? Malt sugar? It tastes different than sucrose, doesn't it? It's not as sharply sweet. It's a little more dull sweet. Yes? Glucose and glucose. Galactose and glucose makes up lactose, which we get in milk. Okay? You with me? All right. Polysaccharide. Many, many, many little monomers stuck together. This is an example of something called glycogen. So, did you guys eat lunch? Okay, good. No lunch? Did you eat breakfast? No, but your brain is so pissed off at you right now. I know. Feed it, will you? So, you're into breaking down as much of this stuff as you possibly can at this point. When I take it in, when I eat, I'm going to use some of it. And we're talking carbohydrates now, right? And I'm going to store some of it. I'm going to put some away in the liver. I'm going to put some away in my muscles. Just for the perfect example of why I do this. Because now I need glucose. I need glucose to send to my cells so I can make energy. If you're not taking it in, I have to get it from somewhere. So I have to do what? I got to break it down. So this polysaccharide is stored form of glucose that I'll break down when I need glucose in my circulatory system. Other things do this, like starch, potatoes, right? Potatoes are examples of big, large carbohydrate molecules. If I take a small little piece of potato and I stick it in my mouth and I leave it there for a really long time, what's going to happen to it? It's going to start to break down. I'm going to use chemicals in my mouth called enzymes to break it apart. And it's eventually going to taste sweet to me, believe it or not. Because those enzymes will break the big, huge molecule into what? Little monomers of glucose. Glucose on my taste receptors will make me think sweet. Okay? So what's another example of a complex carbohydrate? Potatoes, pasta, bread. Where do those all come from? Something that grows out of the ground, right? Who do you think makes these carbohydrates? Plants. Through what reaction? Photosynthesis. Can we make carbohydrates? Nope. We got no chlorophyll. Where do we have to get our carbohydrates from? We got to eat them. We got to eat plants. Yes? So, carbohydrates, very important. We're going to use carbohydrates for making energy. Next semester when we talk about cellular respiration, we're going to talk about the breakdown of glucose. And that's going to give us lots and lots of ATP, and that ATP is going to help drive many reactions of metabolism. The next organic molecule I'm talking about is something called a lipid. Lipids are carbon-containing. They're organic molecules. But what are they made of? Lipids are fats. It's a kind of a same name kind of thing. We have different parts of a lipid molecule. We have some sort of glycerol molecule, and then we have something called a fatty acid. When I look at fats stored in our body, we see them typically stored in our body in this form. It's called a triglyceride. What's a triglyceride? Yeah, a glycerol and 3 fatty acids. Now, how do I get the fatty acids to stick onto the glycerol? What is that called? Dehydration synthesis. So I suck out water to bring the two together. Okay. Another type of lipid we're going to see in our bodies has the glycerol. It has 2 fatty acids and kind of a different fatty acid, one that's kind of a little bent. And it also has another little group on it called a phosphorus group. We call these guys phospholipids. And when we talk about the covering of our cells, we're going to see that those guys cover the whole outside of our cell. That's what our plasma membranes are made of. So we have neutral fats or triglycerides, we have phospholipids, and then we have, starting to get really big, very large lipid molecules. And when anything in chemistry starts getting really big, we don't line it up straight anymore. What do we create? We create this structure. This is a steroid. Just a big lipid, basically. Cholesterol is a steroid lipid. Big. Why do we need cholesterol? I thought that was bad for me. Thought I didn't need that. Look, all cholesterol is just plain old cholesterol. What do I need cholesterol for? You know? Structure. There's going to be a very important structural component of every plasma membrane in my body. It's so important that I make it myself. You know who makes it? The liver. Do I need to eat it? Nope. Because I can use some lipid ingredients from other things that I eat that contain lipids in them, and I can use those components to make cholesterol. Some of the foods I eat have cholesterol in them. Only from what? Only from animals. Do plants have cholesterol in their plasma membranes? Nope. So cholesterol is a very important component of all the plasma membranes in my cells, in my body, animal. And it's a very large lipid molecule called a steroid. So what were lipids made of again? Glycerol and fatty acids. We can have phospholipids if we add a phosphate. Eicosanoids is another name for very large lipid molecules. So what were carbohydrates made of again?
Sugar.
Simple sugars. What are lipids made of? Glycerol and fatty acids. What's the next organic molecule I'm going to be talking about? Proteins. Proteins are carbon containing, just like everybody else in my family here. But the building blocks, the monomers of proteins are carbons in there. The little building units. Remember the building units of carbohydrates were sugars, right? We call the building units of proteins amino acids. Now the amino acids are called that because they have something called an amine group and acid group. So every single amino acid that we're going to talk about with respect to us in humans, and there's about 20 of them, write that number down because that's important, 20 amino acids, they all have this little functional group, the amine acid group. That's why we call them amino acids. So what's different about each amino acid? This little green square here, this R group. So glycine, that first amino acid, has a hydrogen there, but aspartic acid has this there. And glycine has this there. You get the point? So all of them are going to have this amine and acid group. And the 20 differences are going to be in that little R group. So 20 different amino acids are brought together by a bond called a peptide bond. And a peptide bond is what kind of reaction? Dehydration synthesis. So what do? Suck water out, stick the two together. The bond I create is called a what? Peptide bond. So a single building block of a protein is an amino acid. If I bring two amino acids together, what do we call it? It's a dipeptide. So we name it for the bond that brings it together. Guess what a whole bunch of amino acids put together are called? Polypeptides. And these are what? proteins. When we talk proteins, we can get pretty honkin' big. And what we're going to see in the protein world is there are different structures depending on how big I get. The simpler proteins fall in line, straight line. They follow what we call the primary structure. Once I start getting a little bit big, I'm going to steal a piece of paper here. No, I do. Okay, I'll take that one. Oh, no, you wrote on it already. Oh, all right. Give me. All right, primary structure, right? When I start getting bigger than that, I don't want to, yeah, I'm going to start minimizing the space. See what I'm doing? So when I start getting really, really big, I'm going to start folding myself so I fit in the same amount of space. So we have secondary structures. We can either fold it like that, and that's called a beta pleated sheet, or another trick I could do is like make a funnel out of it and fit it in the same place. See? I'm mutilating your paper. So if I fold it up like that, that's a what?
Helix.
It's called an alpha helix. So once I start getting a little bit bigger, I don't want to take up as much space, I start folding myself as a protein. So I can be an alpha helix or a beta pleated sheet. That's a secondary structure. I can get even bigger than that. Now what am I going to do? I'm going to take a combination of the two and I'm going to fold it up really tight. Yes? What's that? That's a tertiary structure. I can get even bigger than that. Blah! I can take a bunch of these and stick them together. What do we call those? Quaternary structures. You want that back? Thank you. Okay? That's pretty much as far as we go. Then we start breaking things apart. So, we have primary, secondary, tertiary, and quaternary protein structures as we get larger. Yes?
Now, like our DNA, does that tend to be more like the secondary structure?
The DNA, when we talk about that molecule, wraps itself in a helical form. Yep. It's not protein, though.
Right.
Yes? So proteins are made of what? Amino acids. Brought together by what? Dehydration synthesis, peptide bonds. What kind of structures can they form? Thank you. Primary, secondary, tertiary, and quaternary. Now, believe it or not, that fold is very, very important, because that fold is going to determine what shape I am. And the reason I fold this way is because of the different molecules I have contained in me as a protein. That folding is very important. Yep. This whole thing isn't going to be with everything. Maybe only this section of that protein is going to be reactive with something. So it has to be in just the right spot, and I have to be folded just the right way in order for me to work the way I'm supposed to. You following me? What can mess up the fold? You probably messed up some folds this morning. Anybody cook yourself some breakfast this morning? Or go to Dunkin' Donuts and get one of those sandwiches with eggs in them? The white of an egg is albumin. It's a protein. If I take it and I throw it into a hot pan, what happens? What do I do to it? I don't lice it, I unfold it. What's the fancy science name for unfold? Begins with a D. Denature. Okay? So heat can be one thing that can unfold this protein. Denature it. Change it. Try to get it back fry an egg. Try to get back to the way it was before. Denatured, it breaks apart the original alcohol. Yeah, it's still a protein. It's still a protein, but is it going to be able to do what it used to do? No. What else can denature a protein? Acids, bases, pH. That's why pH is so important in us, because we make certain proteins that have certain active sites that do very specific jobs. A lot of them are called enzymes. And that shape that we very meticulously made by folding it just the right way is going to help us with chemistry throughout our body, help bring things together or help break things apart. But if I unfold it, is it going to be able to do its job? No. If I mess up the pH in my body, what am I going to do to my proteins? I'm going to mess them up. Are they going to work the way they should? It's very hard to get them back again. Yes? So try to get the egg white back to the way it was before you fried it. Good luck with that, is right. Yeah, you can. But it takes a whole bunch of chemicals, and you probably won't want to eat it after that anyway. So, denaturing proteins, temperature, pH, can mess with the fold. So they talk about some of the different types of proteins in our body and how that fold is very, very important to how the protein is going to do its job. I denature proteins when I mess with pH or temperature. And they also talk about molecular chaperones and enzymes. Again, these are proteins that are going to help with chemical reactions. And basically that fold is going to be a place where chemistry takes place. The other these enzymes, or another name for them is catalysts, do is allow chemistry to take place with a lot less energy being used. If we didn't have enzymes to help us with chemistry, we would have to make so much more ATP that we couldn't survive. Our cells couldn't survive because it would be way too much energy that we would have to produce. So enzymes can help reactions take place and lower the amount of energy it takes for them to take place. So we see them referred to as substances that lower activation energy. Basically lower the amount of energy it takes for chemistry to take place. The reason we're so complex an organism is A, we can produce a lot of energy, and B, we have tons and tons of enzymes that will help our chemistry take place using a lower amount of energy. So without enzymes, many times the reactions don't take place, so they take place so slow, they're not going to be a huge benefit to us. Okay, so they talk about catalysts here and enzyme action in your textbook. We have one more member of our family to talk about. Just a quick picture. This one is on page 53 in your textbook. This shows us how enzymes do their job. So this particular enzyme has a very specific spot on it. Remember my piece of paper has to be folded just right so that that spot can do some chemistry. That spot is typically referred to as the active site. It's going to bring, in this case, two amino acids together and bring them close enough together so that they can what? They can hook up together and form a peptide bond. So when I make proteins, guess who's going to help me do that? Enzymes. When we talk about transcription and translation and protein synthesis with the help of DNA and RNA, we're going to talk about enzymes that are involved there that are going to help hook those amino acids together, form peptide bonds, and form polypeptides. Okay? Everybody with me? All right, so those are my enzyme guys. So who's last? Who's the last member of the family I want to talk about?
Nucleic acid.
Nucleic acids. Now, what are nucleic acids made of? Well, they are DNA, and they are RNA, and they do have carbon, and they do have oxygen and hydrogen and nitrogen, CHON. That's all there. Yeah. So we have little building blocks, little units that we create when we create our nucleic acid. There's phosphate, some kind of sugar attached to something called a nitrogenous base. And somebody said adenine, guanine, thymine, cytosine. Those are the what? Those are the nitrogenous bases we're talking about. The phosphate and sugar create sort of a backbone. And the nitrogenous bases hook up in a very specific pattern. The pattern of your nitrogenous bases is your genetic information. So when I look at the nucleic acid made up of these, what do we call these things? These little units. These are called nucleotides. Anybody ever play with Legos? If I give you 4 different little Lego bricks, what can you make? Anything you want, right? You just put them all in different patterns to make different things, right? What do you think you can make with your 4 different nucleotides in your DNA? Millions of different proteins. And it's based on the order of the nitrogenous bases. So when we look at nucleic acids, they have some kind of phosphate, some kind of sugar. Now this one has a sugar called deoxyribose. So this one is DNA. What does DNA stand for? Deoxyribonucleic acid. So it has to do with the sugar I have here. It's a nucleic acid because it's made of what? Nucleotides. And the 4 different nucleotides I'm going to find in DNA have a phosphate, a sugar, and a nitrogenous base of these 4 different kinds. So the 4 different kinds are adenine, thymine, guanine, and cytosine. Notice their weird shapes. One fits into the other. Do all of them fit together? No. It has to do with their chemical makeup. Adenine and guanine are what we call purines. And thymine and cytosine are what we call pyrimidines. DNA hooks up in what we call a double helix. It creates a ladder structure. So if I'm going to create a ladder, I have to make sure that the bars are equally spaced and the rungs are equally spaced, right? Can I climb a ladder that looks like this? No, not really, right? Yeah, the rungs were kind of like not quite fitting and it wouldn't work, right? Because of its shape, can only match up with what? Thymine. Cytosine can only match up with guanine to create the perfect ladder. And then we're going to take that ladder and do what? Twist it to create a double helical structure. Mr. Watson and Mr. Crick had something to do with discovering the shape. Yep, Rose left out again. Rosalind, who? No. Can't remember her last name. Sorry, bro. So that is DNA. We also have a single molecule that doesn't create a helix. It's got some different nitrogenous bases. It's got adenine, uracil, guanine, and cytosine, and that makes up a molecule called RNA. What else is different about RNA? The sugar. A deoxyribose sugar, it's a ribose sugar. We also have our very, very popular friend, ATP, who is also in this same nucleic acid family. Now, what is the nitrogenous base in the nucleotide that makes up ATP? It's always going to be adenine. The sugar is ribose, and we get a little carried away with the phosphates. ATP has how many phosphates? 3. ADP has 2. AMP has 1. That's what we use for our energy source. We're going to break that apart and use it as an energy source. So throughout this whole book, we're going to see that little sparky looking ATP. And we remember from this chapter that we use ATP as our energy source. When we break off a phosphate group from ATP, it gives off energy. That energy is going to help drive reactions of metabolism. It's going to help enzymes do work in plasma membranes. It's going to help muscles contract. It's going to help chemistry happen. So the energy I get from breaking that molecule apart is the energy I'm going to use like batteries to drive my flashlights, right? What happens if I don't have batteries? Flashlight don't work. What happens if I don't have ATP? Your body don't work. Chemistry doesn't happen, right? So ATP is our nucleic acid friend as well. All right, so what are the 4 organic molecules? Carbohydrates, lipids, proteins, nucleic acids. What are carbohydrates made of? Sugar, simple sugars. What are lipids made of? Glycerol and fatty acids. What are proteins made of? Amino acids. Brought together by what kind of bond? Peptide bonds. And what are nucleic acids made of? Nucleotides made of phosphorus, sugar, phosphate sugar, and some kind of nitrogenous base. Yes? DNA has how many different nitrogenous bases? 4. RNA has how many different nitrogenous bases? 4. Different sugar, right? Ribose, deoxyribose. We good? Chapter 3. Any questions? Is the sign-in sheet going around? It's all done. Anybody not sign it? Good. Okay. Let's start chapter 3. Chapter 3 talks about what?
Cells.
Cells. So we're going, see, we're kind of going in order here, right? We started off with atoms and elements. Now we're into, well, and then molecules kind of. And now we're into what? Cells. Cells is the smallest what? Living unit. Are all cells the same? No. They have all the same basic parts, but they'll have different functions. They'll carry on different chemistry. They'll make different things. We are multicellular organisms. So we have a whole bunch of different types of cells that are going to do a whole bunch of different types of work for us as humans. So cells a basic structural unit, basic living structural unit. And when we look at them under the microscope, we see them very different in shape. Again, cells can connect things in the body. So when we talk about tissues, which is the next chapter, we're going to talk about tissue groups of cells that connect things like connective tissue, or cover things like epithelial tissue. Some cells can move parts. What kind of cells are those? Yeah, muscle cells. So they can move different things through different organs or move you from one place to another. Some cells store nutrients like adipose cells. Some cells can fight off disease, recognize foreign invaders. When we talk about the immune system next semester, we'll talk about those guys. Some cells can send messages from one place to another. When we talk about the nervous system, we'll talk about those cells. And some cells are kind of what I refer to fondly as half cells, contain half the genetic information they need so that we can create new organisms as a species. And we call these guys reproductive cells. We'll talk about them at the very end of second semester. So not all cells look alike, but all cells have the basic same structure. On page 63, figure 3.2 in your textbook, where are you going to see that again? In your lab exam. So you should be able to know all the parts of what we call the generalized cell. When we start talking about different systems and different cellular material, we'll see every cell looks different. Some cell might have more of these parts than others. Some cells, very rarely, are missing parts. So when we talk about a cell, these are the basic structures in the cell. We have some sort of covering that goes around the outside, and then we have a whole bunch of little parts inside, and those little parts are called organelles that form different functions for the cell. So the first thing the book talks about is what we call the Fluid Mosaic model. This is basically what your plasma membranes look like. So if I take a cell, is it going to be a hard thing? No. It's going to be kind of like runny jello-ish. And like a jelly. Yeah, jellyfish is a good example. Okay, it's not a hard thing. Things can come in, things can go out. But not just anything can come in, and not just anything can go out. So when we discuss the fluid mosaic model of the plasma membrane, this picture here in your text does a nice job of showing us what we're going to find in a plasma membrane. Most of it is going to be these little things that look like clothespins. Now these guys are lipids. These guys are lipids with a phosphate on them. What do you think we call them? Phospholipids. And kind of a two-sided little sandwich kind of thing that goes all the way around the cell. It's called the Phospholipid Bilayer. Now Now, the reason it lines up like this is because part of the molecule is actually hydrophobic, or what we, in layman's terms, afraid of the water. And part of the molecule doesn't mind being associated with water. It's hydrophilic. So guess where the water is here? Yeah. I'm going to connect them all because I feel like drawing them all. But there's a whole bunch of water on the inside of cells and on the outside of cells. So the hydrophilic ends touch water. The hydrophobic ends hide and hang out with each other. The other thing we're going to find here is these orange things, yellow things. What do those look like? Cholesterol. So that's another very important structural component of the plasma membrane. So we have phospholipids, we have cholesterol, and then we have these big purple blobby things. What are these things? These are proteins. Now you notice they don't all look the same, right? A whole bunch of different shapes. Some of them have things stuck to them. They're all kind of strange looking. Well, those proteins all have different jobs. Some of them act as bridges so that things can pass from one side of the cell to another. Some of them act as cellular markers, sort of like street signs to other cells, telling them who they are or what I do for a living as a cell. And some of them are going to perform different chemical functions within the cell. We have proteins surface, either outer surface or inner surface. We call them peripheral proteins. And then we have proteins that kind of hang out in the sandwich there, that phospholipid, called integral proteins. We also have proteins with carbohydrate groups attached to them. Guess what we call those guys? Glycoproteins. We have lipid groups attached to proteins. What do you think we call them? Lipoproteins. So, whole bunch of different proteins, whole bunch of different functions. As we go through the course, we're going to talk about some of those different functions. But that's what they describe under membrane proteins in your book. So we have ones that go through the membrane or ones that sit on the surface, outer or inner, integral proteins versus peripheral proteins. They talk about what we call the lipid raft. 20% of the outer membrane surface proteins are what we call lipid raft. And this is what, because of the chemical structure of these guys, this is what's going to be the group of molecules that are going to determine whether you can come in or not. With the help of proteins, I'm going to determine whether you can come in or not. Lipids usually don't have, believe it or not, even though lipids are large, tend to be large molecules, they can come in and out a little bit easier than something that's water soluble. Because remember the hydrophobic ends of my phospholipids? Where are they hanging out? In the center there. So without those proteins to help things come across, things would have a hard time going from one side to the other. The next thing they talk about are some of the proteins that are on the surface and some of their jobs. Some of them transport, help things move in and out. And we see this on page 66 in our textbook. Some of them will change chemically and cause chemical reactions to occur within the cell when they come in contact with other things. So when we talk about hormones, for example, we're going to talk about these receptor proteins. When the hormone comes in contact with it, it's going to cause a chain reaction to occur within the cell and cause changes in what's going on chemically. Those are called receptor proteins. Some of them are going to attach one cell to the next. Some of them are going to attach fibers on the inside of the cell to give it its shape. And that's what they talk about when they talk about attachment to the cytoskeleton or extracellular matrix. What's extracellular? Outside the cell. When we talk about tissues, we're going to talk about not just a cell, but a group of cells that are what? They're all stuck together for some kind of purpose. So that's what these guys are about. Some of these proteins are going to carry on other chemical reactions, and they have enzyme-like activity. So they can help bring things together or pull things apart. Again, the joining of two cells might be its job in intracellular joining. And then some of them act as little street signs. So how do I know a cell is foreign to my body when it comes into my body? How does my body know? That's a bacteria, and it's going to make me sick. Yeah, those little cellular recognition proteins aren't just exclusive to you. All organisms have them. So I know, my cells know, that something is foreign to it when it comes in because it does not recognize the proteins on its surface. So all living organisms have cellular recognition proteins on the surface. Your cells are taught to recognize your proteins. And anything that's not yours is considered what? Foreign. Yes. What about an autoimmune disease? Is that what you were trying to ask me? So what happens what happens to us when our cells can no longer recognize self? They start attacking our own cells. That's called an autoimmune disease. So something goes wrong in the recognition process, and the cells can no longer recognize self-antigens. Is that, excuse me? Correct. Something goes wrong with the cell's ability to recognize those cell recognition proteins, and you start attacking your own cells. Is that normal? No. All right, they talk about cell junctions. Holding cells together is very important. We are multicellular organisms, so the junctions between cells are extremely important. They talk about several different kinds in your textbook under cell junctions, page 66. First one, they talk about something called a tight junction. It's kind of like Ziploc. You know, the two sides fit together, hold the two sides really tight, and you hope that they're good and zipped if you have a bag of angry bees, right? Tight junctions. They're going to help fuse cell membranes together. Desmosomes add a little extra tightness to the Ziploc. It's kind of like a little spot weld. Ever look at Velcro up close and personal? Yeah, that's what it's kind of like. So we have little proteins that kind of hook and link to give extra added little tightness to cells. In cells, for example, that make up your bladder. What's inside your bladder? Urine. Waste. Do you want your cells leaking? Do you want the space between those cells to not be very, very tight? No, because what will happen? Urine will come out into your body cavity. So in places that need a very tight fit, a little extra something with desmosomes. The next one they talk about is a gap junction. When we talk about nerve impulses and chemistry going on to cause changes within cells, we talk about chemistry, chemicals moving from one place to another. Now, is everyone's heart beating in this class? Hopefully, right? And what is it that causes our hearts to beat? Do you know? Muscles contract, and you know what causes muscles contracting? Or changes in what? No? Chemistry, chemical changes. Now, my heart muscle has to constantly beat. It has to shorten and lengthen, and shorten and lengthen, and shorten and lengthen. 24/7, right? So constant chemical change has to happen. Some of that's caused by the nervous system, but some of it is caused by communication from one cell to the next, passing the chemical change along from one cell to the next. Are going to help that sort of communication happen. They're called gap junctions. So it's kind of like taking a straw and poking it through the plasma membrane of two neighboring cells and allowing chemical changes to go from one cell to the next. There's a group of cells we're going to talk about when we talk about the circulatory system called Autorhythmic cells. And these are going to pass a wave of chemical change from one these little gap junctions. Okay? That's probably a good place to stop. We'll pick up with plasma membrane and membrane transport on Wednesday. Yep, because we finished, so glad you brought that up. We finished chapter 2. So what am I going to do when I leave you today? I'm going to go back to my office. I'm going to open up the chapter 2 quiz. And what are you going to do? You're going to take it before when? Before class on Wednesday.